Assalamualaikum and a very good morning to everyone. Today I'll be presenting a review paper on marine microalgae Salina and N.Occulata as potential sustainable nutritional food source. My name is Nohanisa Fateh binti Ruslan and I'll be proceed to the outline of my presentation. This will be the outline of my presentation, starting with introductions and end up with gunshots and references. Alright, so I will introduce to you guys about my topic. So what is microalgae? Microalgae are unicellular photosynthetic microorganisms that live in saline and freshwater. They both be able to survive in those conditions, and they are converting the sunlight, water and carbon dioxide to algal biomass. This mentions by De Vera et al. 2018, and this species also contain a high nutritional value, such as high in protein, plant-based vitamins, minerals like potassium and magnesium, pigments such as carotenoid, omega-3 and omega-6 fatty acids, and antioxidants. As you can see from the picture sourced by Machado et al. 2022, mentions 5 major components of the potential sustainable food sources in the field. For example, as you can see in animal feed, emulsifier, food additive. Same goes with the pigment that will be able to contribute in a cosmetic field, dye and paint, and also in a food supplements. And last but not least, in lipids. Lipids has the biofuels, the cells, and the glycerol productions. The problem statements of my review study would divided into three, where first is the global human populations as the number increasing at an incredible speed over the years, with the estimation around more than 70% food productions required in accommodate with the human population growth by around 2.3 billion around the years 2050, as well as the global hunger that mentioned by Chia et al. 2019 will be one of the main cause of my review paper. And next is the global warming itself. We all do know as the human increasing, technology increasing, and the earth is getting old. So the global warming effect of the food stock, food feed, food crop these days contribute to a lot of carbon dioxide emissions. By means, the microalgae will be able to emit around 3% of them. And lastly is this study is compositions of the Salina and N.Occulata as becoming sustainable food source has not been documented well and understood. The significance of study will be revolved around the literature review, provide information on the possible substitutions of current food sources with microalgae of N.Occulata and Salina, along with understanding on their parameter applicable toward growth of the species. And last is the study enhance the food science and the food technologies to find novelty productions in food industries and utilize the microalgae components according to the specification requirement of microalgae species in resolving future food crises and using the global warming effect. Correlate to the significance of study, the objective of my study will be to comprehend the components of N.Occulata and Salina microalgae as probable potential future food sources that correspond to its cultivation parameter, and to identify the application of N.Occulata and Salina microalgae in food industries. My study will be focusing on how the microalgae be able to become a component of potential sustainable food sources, and what parameter measure that affect the compositions of nutrition upon the microalgae species. In continuations to literature review, so the species I've been studied will be Salina, which is a marine species that high concentrations of protein, plant-based vitamins, minerals like potassium, magnesium, and antioxidants. This species has a very interesting facts where the cultivations of the species able to produce high concentration of beta-carotenoid, which is the source of antioxidants. It able to grow in the conditions of extreme salt water conditions that optimally around 10 to 15% of salt, which allow them to easily grow without any much competitor, as they've been temperature required around 5 up to 40 degrees Celsius. N.Occulata, also a marine water type microalgae that rich in protein, carbohydrate, vitamin, antioxidants, and particularly polyunsaturated fatty acid, which is a omega-3. The characteristic of the greenish phototrophic unicellular non-zoospore productions, free-floating species with a diameter of 2 to 4 micro and a non-azenic growth in a temperature range of 11 to 16 degrees Celsius. The bioactive component. Bioactive component is a chemical that contained in the plant itself, microalgae, that be able to be beneficial to the human body. So, for example, can be said as protein, peptide, fatty acid, chlorophyll, carotenoid, and vitamins that be able to be promising for the pharmaceutical, food additive, cosmetic, and biofuel field. The example mentions by Fitriani et al. 2019 about the narcolepsy species, they focusing more on exploring the cholesterol lowering activity level component. As I mentioned that the N.Occulata has high in omega-3, so the species itself able to have around 4.38% omega-3 concentrations of EPA and also DHA. Besides the ZETAL 2021, these Salina species mentioned have high in beta-carotene compositions that act as photoprotector and antioxidants upon synthesis. This photosynthetic microalgae can synthesize up to 10% of dry weight bioactive. Next is their potentials at becoming a sustainable food sources. So here we are going to digest on the nutrient compositions that contain in the D. Salina and also in N.Occulata. Both of the species able to contribute in a high amount of protein. So for the table 1 on D. Salina species, the carbohydrate and lipid is also promoted in a macronutrients compositions as well as potassium, sodium, bicarbonates, magnesiums, and iron. For table 2, the nutrients of protein is the highest with 31.68 and continue to the ash content, lipid, and with carbohydrate. This additional analysis were done upon the N.Occulata species as they have a lot of potentials as fatty acid component with the test of phytotocopherol, phytosterol, and also an analysis on omega-3 and 6 by the HPLC-GC. And as you can see, the B-citrol, stigmasterol, and omega-6, omega-3 results in quite high number of percent. Is upon their cultivation systems. So in cultivating the microalgae is non-arable, where there's not required any land or either fertilizer. It's required a space for you to cultivate itself. So they divided into 3 types of cultivation mode, which would be photobioreactor, which is a closed systems, and a raceway pond, which is a open systems, and hybrid system is the combination between both of them. As you can see from the graph that mentions by Narala et al. 2016, they compared their growth rate and also biomass productivity based on different based on different cultivations method. So all these cultivations have a significant difference of P less than 0.05, where we can see that both hybrid system and in their growth rate as well as biomass productions able to produce a high number compared to other two method. This is particularly because that the photobioreactor and raceway pond are combined, where in the earlier stages where the closed system photobioreactor is where we contribute on specific parameter that we decided to do. We can enhance their nutrients, we can do alterations in their parameter growth, so that, and we decided on the cultivations algae requirement that we want, and proceed to the raceway pond where it increased the biomass agar. So as we able to not only produce a great microalgae but also a high quality and quantity by hybrid system. Alright, to go in depth on their parameter only can be adjusted or altered by the photobioreactor, which is a closed system. Cultivations process can be alterations of macro and micro nutrient supply during their media, as well as their range of temperature, mixing conditions, nutrient compositions, light intensity, pH concentration, and gas exchange during their aerations processes. All of these may affect their biosynthetic pathway and may able to produce different bioproduct of a batch of culture. And based on my reading, the efficient way is by modulate stress upon the culture media, and the cultivation of D. Salina and N.Occulata shows significant increase in their biomass production as well as their growth rate. So as you can see, here is a table that I've been gathered based on their microalgae species, culture systems, nutrient media, environmental control parameter, outcome, and sources by. So as you can see, the culture system of course will be closed system as the parameter been adjusted. So as you can see, the nutrient media compositions for the first point, the SMG media been using the same media, but the manipulative variable in this experiment would be the abiotic stress conditions that been included in their culturing system, where use high light intensity, nitrogen depletions, and high salinity. Both three results in different outcome, where for both high salinity and nitrogen depletions, the conditions significantly inhibit the cell growth. However, nitrogen depletion significantly induce the beta-carotene accumulation, and for the high light intensity promote the lutein production, which is the black pigment. So next is the using of walnut media and also walnut media that added with nitrogens, phosphates, and potassiums. The manipulative variable will be the light intensity, where we use blue and red LED, and it results in a high Salina concentrations for the red LED treatment. And next, for Salina, the photobioreactor undergoes three different nutrients media, which is F2, which is the original OG, what we call as the compositions of nutrients, and modified Conway as well as Johnson's media. So the constant variable will be the aerations, the artificial seawater, and constant flow rate. So the media introduced the maximum accumulations of pigment, especially violacetine, and chlorophyll A can be observed by Johnson media. For Conway media, shows the highest level of total lipid, which is 43%. And for the N.Occulata species, use Gillard F2 media with a different concentrations of illuminations light for 3,000 to 7,000 lux. So based on this, results outcome of light intensity at 5,000 lux achieve high growth rate with a shorter generation time of 2.9 days in within range of 3 to 5 days. Continue to the next table, which is species of N.Occulata. Been using the photobioreactor annular, the F2 media is constant with manipulating variable of their inorganic nutrient, which is the concentrations of bicarbonate solutions are divided into 6 cultures concentration, which is 0, 6, 15, 30, 42, and 60. So the outcome changes of the optical density around time period of 72 hours, and the highest in the culture supplement with the 60% 60 millimolar bicarbonate solutions with growth rate of highest of 0.5 compared to the conditions of control, whereas not really much inorganic changes of 0.330. Next is walnut media composition used with manipulative variable of 3 different concentrations, which the media itself been manipulated, where the walnut media first is the walnut media, second is the medium that is nitrate concentrations of 50% less than the amount from the control, and another one is 50% more of phosphate from the control media. So the outcome within 120 hours cultivation period shows the reduced nitrate, which is the sample that have less 50% of nitrate compositions have low dry weight. However, it high lipid concentrations of 25.77%, and a sample that has added 50% more phosphate contribute to a highest growth rate of 0.048 gram per liter day. And lastly is the modified walnut media used with manipulating variable of the natural light and also blue light. So as you can see, natural and blue light emit a lot stronger emissions of wavelength, thus it contribute to more than 70% increase in cell growth component in their cell density, wet weight, biomass productivity, and also lipid content concentration. Last but not least, their potentials are applicable in the food industry itself, where they're rich of source of basic nutrient component such as protein, carbohydrate, mineral, vitamins, and easy manipulations of their compositions according to desired compositions by the market. These morphology of the microalgae itself is permeable membrane, so they'll be able to use and able to modify a little bit of their pathway so that we can get the desired composition that we want. So the marine microalgae as N.Occulata of green colored suspensions of microalgae has been applied in a dietary supplement of source as omega-3 mentioned by Kagan and Matulka 2015. They done a toxicity analysis with the on upon the 10 species of male rat and 10 species of female rat. This species has been giving the omega-3 orally, and they've been analyzed every 3 days, and particularly there's no sign in their kidney, no sign in their blood samples of any toxicity effect. And next is food production such as pasta were commercialized with the additions of the microalgae color and flavor mentioned by Muti 2016, and the applicable of food additive of Salina upon as emulsifier resistance to the oxidations reactions thickener physiological structure of food that assimilate in the food productions along with coloring agents in bread as protein polysaccharide mixed gels that enhance the dough structure. As you can see the image at the slides where this Salina has becoming a coloring in food, and as well as N.Occulata in spaghetti in the noodle itself. For my suggestions and recommendations, so in a nutshell, the review has revealed that the species of Salina and N.Occulata able has the potential as sustainable food sources. The mechanisms and parameter control cultivation able to produce a desired amount of nutrient content of microalgae, which is bioxidative stress, where these Salina components are more drawn to the beta-carotene properties as antioxidants that designed in a functional food for a prolonged shelf life of product. And for the N.Occulata species, their morphologies and phytochemical properties has potential in high lipid, where a major of fatty acid of omega-3 and omega-6 that be able to lowering the cholesterol level of the human. So however, the recommendations I would like to suggest is the more study need to be done towards the species in food industries, as it in encountering the limitations of its applications in food, such as becoming natural replacement of synthetic additive, and discover of many more potential in utilizing this species. As we all know, the cultivations might cost a fortune because they are still in a pilot test. That's why I recommended a lot more research done commercially in finding the most efficient way to cultivate, so that we be able to commercialize these products and be beneficial to humans. Here is my Gantt chart, and this is my reference that we use for these slides. That is all from me. Thank you.